Thank you, everybody, for coming. I'm especially happy to see lots of our newer members joining us today, including our current speaker, who's one of our speaker, Paolo, who's one of our newest members as well. So fantastic to see lots of new speakers. And if you're not a member, I will be putting some information about Validate in the chat so you can join and be able to meet people like me and Paolo, etc., in person at the upcoming events, etc. My name is Blakely. I'm the Communications and Events Coordinator at Validate. And today we have two excellent speakers. Our first speaker is Paulo Antas. He is a professor of biology and a senior researcher at Fiocruz in Brazil. He is one of our most recent Validate members. I think he only joined about a month ago. Paulo's research focuses on innate immune responses to TB, and he specializes on cellular immunology of TB vaccines, amongst other things. He also studies things like in vitro immune response, pre-BCG vaccination, and inflammatory cytokines. And so he has quite a broad, he's going to be bringing a lot of expertise to the talk today. And our other speaker is Thales A. Campello. He is a pharmacist and PhD student at the University of Ceará in Brazil. He specializes in clinical microbiology, and has worked on things like multiplex PCR, nested PCR, and is currently a PhD student. I think he works with Paolo also. So yes, I'm going to hand over to Paolo after my waffle. Sorry about that. Who will start the presentation. Thank you, Paolo.
Yeah, thank you, Blackley. Thank you, all the people that are listening to us today. I have to make a special I'm very thankful for the Validate initiative, particularly Blackley and my colleague Mario Flores Valdez from Mexico. They invited me to become a member of this initiative, so hopefully we can share a lot of thought about not only BCG, tuberculosis, or other kind of disease that I think is going to be very helpful for all of us. So what I'm expecting from the talk today is coming from you guys, that will give us feedback from what we are now and what we think that the path that we're going to follow. So after that, all the thing that I want to acknowledge is Professor Campello from Universidade of Ceará. He accepted my invitation to also share this presentation with me. He has a lot of background in molecular biology, and he going to tell us about the prediction that he made using an in silico analysis for some epitopes that would be useful to, fortunately, in the future, we can insert in the BCG and make a better vaccine, not only for BCG, but also against COVID. So I'm trying to handle these two presentations at the same time to see if I cannot take too much time to present to you guys. And I'm very looking forward to our Q&A section, because I want to have a feedback from you. So as Blackley just said, we are coming from two separate institutions in Brazil. One is Fiocruz, where I'm particularly have a position, but I'm also getting up another position, a provisional position at the University of Ceará, where Thales is located now. So I'm trying to handle these two positions, but the things that we are looking for the same background in terms of what we want to do, that is doing research, clinical research, or in vitro studies using BCG as a main target. So I'm trying to cover the two aspects, and let's see. Okay, so just non-disclosure, and what I expect from today's talk is to give you guys a background. I hope I can share this screen. Okay, I can give you a background about what we are now in terms of BCG heterologous protection. We have, as you may know, a lot of strains that cover let's say, several vaccines, that I could say like that, because BCG is not only one vaccine, we have several strains. all over the world. They can behave as different from another. We try to show you that. And then I introduce you our cohort that we try to cover some aspects of the in vitro immune response. From there, I will go to show you, try to convince you that we have different cell death patterns using different strains of the BCG, plus the biomarkers that they're able to produce in vitro. Then Professor Campello will talk a little bit about the prediction of the epitopes, and then I try to wrap up all the information and we can open for discussion. So I like how to start, get started this presentation with this simple statement, because most of the students, when it came to my lab, and the first thing that they asked is how much do you know about a vaccine that just turned in a century? It's a centennial vaccine, and why we don't know too much about how the vaccine works. So I tell them that we're still studying. It's it's a centennial vaccine. There's a lot of work to do, but maybe we don't know why the new vaccines against TB don't work, because we still don't know how much the BCG should protect given vaccine. So it protects against several other forms of TB, particularly the severe ones, but the one that is spreading all over the globe, that is the pulmonary form, its protection is waning against the time, and we don't have too much protection. So we don't know why that happens, surely, but we are trying to focus on that. It's not what I'm covering now, but it's this aspect that I want to address. So what we've seen, particularly myself, when I start reading about the BCG vaccine over more than 20 years ago, is that a lot of things have been done by them about trying to evaluate new vaccines that maybe could replace BCG or get more insights about the new vaccine. However, the BCG vaccine itself, it carries a very good protections in terms of it's a good carrier that would be used since it started to become a vaccine to be promoting like an heterologous effect. So what I want to talk about that is that since there is an adjuvant vaccine inside the BCG vaccine, because the vaccine itself, the BCG itself, it can be used as an adjuvant. Why don't we can start introducing new candidates for heterologous protection? I'm talking about the virus antigens, or talk about parasite antigens or the bacterial antigens that can also give good protection, plus protection that the vaccine can afford against tuberculosis. So because of that, maybe a lot of study came. Yeah, a lot of study came to try to do that. So trying to generate recombinant BCGs that could serve, the BCG itself, as I told you, could serve as a good vector for other infectious disease. And one of the focus that can gain a lot of interest lately was against viral infection, particularly COVID. So there is some work that has been done to try to see if the BCG itself can also carry some antigens that protects against COVID. However, there is no success, as far as I know, about using the recombinant BCG for protection of heterologous disease, but maybe we are a good way to find it. So there is another thing that I just told you about the BCG itself, the heterologous effect protects against other diseases, not only tuberculosis. And the reason of that is that the data that came from the literature about the reduced neonatal mortality, particularly those neonatal, those kids that they get fatal respiratory infection and sepsis, they were a little bit protected when they got vaccinated with the BCG. So that's the first things that they came talking about an agnostic of target effect of the BCG vaccine. Some of the works that were done in kids, just pick some of them. The literature, it's a clinical trial that was done in Africa using, as I told you, there are a lot of strains of BCG, so they use a BCG Danish. And when they gave BCG Danish to low birth weight babies, it's reducing neonatal mortality by almost a half. Also in Spain, they also used BCG Danish in another trial. They saw that there is a reduction in the hospitalization for those kids, the reduction about 32% for respiratory infection and 53% against sepsis. But more remarkable is this work that was done by Roth and colleagues in 2010, that when they gave BCG as a revaccination plus a booster dose of diphtheria, tetanus, and pertussis vaccine, it reduced, I think it's a remarkable 64% the mortality, all-cause mortality in those kids. So there are two explanations for that. The first one is the cross-reactivity that BCG has induced in the T cells against the other pathogen, and also the train and immunity. So not only in kids, but also in our seniors, it has been shown to protect against respiratory infection. There is a clinical trial that uses intradermal BCG. They protect the elderly from respiratory infection, and they increase also the responsivity of the cells from circulation when they use an unrelated stimuli. As I told you, there is also a study using model of BCG, an idea about BCG to protect against COVID. So during the pandemic, it was suggested that the countries where they have campaigns, BCG vaccination campaign like Brazil, that campaign, that vaccination coverage would be associated with reduced number and severity of COVID. But more recently, another study showed that it seems to be ineffective. The BCG vaccination seems to be ineffective against COVID-19. So there are some statements about these heterologous effects for the BCG vaccine. First of all is that maternal priming using the BCG increased the beneficial non-specific effect. Also, the BCG revaccination, booster of the BCG, seems to have a more direct effect than the priming vaccination. Then a colleague of mine, Frank Chan from Australia, they came with this statement that I think is very remarkable. That's revaccination with the BCG, it increased the beneficial non-specific and specific effects of BCG vaccine, and that kind of effects, it's relying on age. As I told you, the streams of age seem to be more protected, and also the gender, since girls seems to be more protected than boys. So because of the pandemic, lately, what we've seen, particularly in some Latin American countries, there was a disruption in the BCG vaccination coverage, particularly in the childhood. There is one study that was done in Brazil showing that during the pandemic, there was less coverage, it's a negative coverage of the BCG. They start to recover just a little bit after that. But one thing that came from this pandemic situation, it reinforced the worst scenario about the BCG adherence, and the process seemed to be more dynamic and factorial. So there is one fact that came from the COVID-19 pandemic, is that the delay in the BCG vaccination during the pandemic, it led to higher pediatric mortality from TB. So conducting vaccination catch-up could mitigate this impact. So let me explain, so for some of you that is not familiar with the BCG field, as I told you in the beginning, there are several strains that were spread all over the world since the vaccine was discovered. by 1921. By 1924, several lots of the vaccine came from Institut Pasteur in Paris, and it started to spread all over the world. And each laboratory that got one of these vials started to grow the BCG by their own way. So after 50 years, it generated a lot of difference in terms of genetic and also biochemical background. So after that, the community were aware about the difference between the different strains that happened. The first lot was spread. So Broch and colleague, they decided to mount this genealogical tree to make the things a little bit easier, because it's a little bit difficult to understand what's going on in terms of the spread of the difference between BCGs, among the BCG strains, sorry. So what was very clear by this genealogical tree is that there was one of the regional difference called RD2 that happened in about 1927 that clearly cut the genealogical tree in two, giving an idea that there was early shared streams in the bottom of this slide, and also late shared streams about in the bottom of the slide. So there is a clear cut about that. I won't talk too much about details about that in genealogical tree, but I just want to call your attention that we'll be using strains from those two different, let's say, separation that they made early and late streams. So after that, what WHO used to recommend is that because of those different in terms of the strains, studies should be done to test the efficacy of those different BCG vaccines. So one of the things that clinical trial used to do is looking for the scar that the BCG used to give for someone for the vaccines. So looking at the scar, you can have an idea if the patient got the scar, got the vaccine, the patient doesn't got the scar, but even though they got it, she or her got the vaccine. So what we are looking at is the number of the people that got the scar and doesn't got the scar, looking at what kind of strain can give or not the scar. So one of the studies that was very important in terms of looking at the scar was done in Africa, where they looked at the scar in children that got BCG Russia, particularly in Guinea-Bissau. They saw that in Uganda as well. So they got children, when they give children the vaccine, the BCG Russia, almost half of the kids got the scar. On the other hand, in the same area in urban Guinea-Bissau, when they gave the BCG Danish, 72, almost all the kids got the scar in that. The same thing was done in Brazil very recently when they look at BCG scar in kids that got the BCG Russia and those that got the BCG Morro. Let me explain why that happened, because BCG Morro was given in Brazil as a vaccine until 2017. And after that, the ministry decided to change the strain to BCG Russia. So they start to make this study because they want to see if there is any correspondence in terms of BCG Morro and BCG Russia in the Brazilian population. And there was a difference in terms of scarring, and I will show you later a difference in terms of in vitro immune response. So it was clear by that time that the BCG Russia, because of those studies, that BCG Russia seems to is less likely to produce a scar than both BCG Danish and BCG. What about TB risk? The risk to acquire tuberculosis. So there was one study that was done in Hong Kong that showing that the risk for acquiring tuberculosis using BCG Pasteur was almost a half less than using the BCG Glaxo. And in Kazakhstan, this kind of risk to acquire TB was reduced by almost 70% using BCG Tokyo, almost 40% using BCG Serbia, and 22% after BCG Russia, once again showing that BCG Russia doesn't seem to be a good advisor. So the conclusion from those studies that different strains are likely to have different specific and non-specific effects. In order to try to contribute to this field in terms of gaining insights about different BCG strains that happen in the world, we're trying to not only recognize there are some clinical trials that show this different in terms of strain, but we also want to see if that kind of difference would also be translated to, for instance, in vitro models. So after we do those studies, and particularly when we start using several strains of the BCG, we look at for different, I would say, outcomes. I don't like the word, but it's what I want to say is that we look at for different ways to check for the immune response. So one of the ways that we look at was cell death patterns in terms of different strains and also some biomarkers. So what we have done so far is recruiting healthy adults in Brazil. They have been vaccinated with the multiple strains, as I told you, until 2017. They were vaccinated because in Brazil it's mandatory. They were enrolled before 2019, before the COVID pandemic. Since vaccination in Brazil is mandatory, it relies on individuals that are naive against BCG, not only BCG, but also against mycobacteria. We should look at individuals that never got any kind of contact with those antigens. Then we have to recruit newborns or their cord blood cells. We drown the cord blood cells from those individuals from the umbilical vein, and then separate the cells and make the same kind of study that we have done in the adults. And the other group there I want to call your attention is those adults that they are enrolled, that have been enrolled after COVID-19 pandemic. This is a work in progress that Professor Campello is conducting there in Seattle. There are some exclusion criteria, like individuals cannot be HIV, cannot have a history, any negative, there's no history of malignant or TB or degenerative or transmitted disease, no diabetes, no use of corticosteroid at the time of the study. Because this is a study that recruit individuals in the blood bank, the identities should be omitted, so there is no kind of comparison in terms of demographics in those individuals, and the study has been proven in several IRBs. So after collecting the blood, what we have done is taking the blood cells, particularly the PBMCs, from those individuals. We infect them with the BCG after several time points. We also use different rates, 2 bacteria to 1 whole cell for the infection. As I told you, for the apoptosis, we use the flow cytometry to check for cell death, particularly in the monocytes. We also have a chance to look at the biomarkers, soluble biomarkers, using multi-array ELISA, and particularly what I'm talking to you today is most data that we collect using cytokines that are produced mainly by monocytes. So to look at the cell death pathway, that will give you some small background about how the way the cells used to die. So there is a clear cut in terms of being inflammatory and non-inflammatory in terms of cell dying in the tissue. A very common way that most of you have seen and have listened to is the apoptosis, the way the cell used to die. And it's a very natural, let's say, natural way that the cell die. And it does not cause any kind of inflammatory milieu there in the system, in the tissue, sorry. And also there is another way that is called necrosis, mostly necrosis, that have been shown to induce inflammatory secretion of, there's a secretion of inflammatory mediates because cells used to leak after necrosis, and most of the content inside the cells used to escape to the milieu, and that we put more cells in at the end, it causes inflammation. So there is this kind of way to explain you. Of course, there is a lot of, particularly in terms of necrosis and apoptosis, that I cannot cover now. And also there is another ways that the cell can die, not only those two, but I think it's more easier for us to understand what I want to show you later. So the dead cells, naturally, when they die, they are clearer from tissue because we have host phagocyte, is homeostasis way to get rid of them. In terms of apoptosis in TB, Fratazzi and colleagues, they study using the MTB bacilli, and they came up with the idea that the virulent tubercle bacilli, they have an evasion strategy in the host that creates a safe environment, and this is suitable for intracellular application and colonization of the cell. This is a kind of defense mechanism that is the MTB can evade from the host, so it particularly it inhibits the apoptosis. It can create a good environment for replication. And to boost the induction of the specific immune response against TB, what the group did is what they used to happen is that to increasing apoptosis, increase the way the cell die by apoptosis, this could be detrimental to the bacteria, then create not a good environment for the bacteria. And by this way, what people used to see, people that study cell death used to see is that this danger signal that are released during necrosis, it can initiate inflammatory response that could be very positive to the bacteria because it eases them to escape the immune response and also create a good environment. So we came up with this statement that I like very much, that guide us in the future of the studies, that most of the time apoptosis it triggered by attenuated non-violent bacteria such as the BCG vaccine, and by this way the BCG plays significant role in shape the immune response to the direction. There is a lot of information in the sentence, but I like them too much because there is a clear cut in terms of separation. What the attenuated mycobacteria does and what the virulent bacteria does in terms of escaping or inducing immune response. To make it more clear, I just came with this slide to just remind us that if we increase the level of apoptosis, it means a pathogen clearance, that what we expect is the BCG vaccine, for instance. But on the other hand, when we look at pathogen persistence, what we can see is high level of necrosis and low level of. So in other words, appropriate cell death pattern is important for a protective immune response. So we came up with after that, we came up with the rationale that if you want to reveal critical aspects of doing vitro cell death profile related to using BCG infecting mononuclear cells, we use it for that study. We use a community-based and open-label cross-sectional population study in Brazil, and we directly compare newborns, those that are naive against mycobacteria, and the adults in Brazil that have been vaccinated. We use the flow cytometer and multiplex to check for those cell death rates and also biomarkers. For the cell death study, what we have done is the flow cytometry. Let me try to explain you what the strategy that we have done. So since we are dealing with PBMCs, we have lymphocytes and monocytes in the culture. For the analysis, what we have done is excluding the lymphocytes and only acquiring cells that are we assume to be monocytes. And then we stain cells with Nexin-5 fluorochrome and clopidogrel iodide. And most of the cells that are apoptotic, they used to reach this region, and the cells are in necrosis, they used to stay in this upper region. So we collect this data, and then what we have seen is that very early when we start using this model is that those adults that have been vaccinated with BCG, when we infect them for 24 or 48 hours, they have a high level of apoptosis. On the other hand, what we've seen in the neonates, in the cord blood cells, that most of the cells, they die when infected by BCG by necrosis. Not only cell death, as I told you, we also look at the cytokines. In adults, we could see that there is a high level of TNF and island beta production, pro-inflammatory cytokines, when the adult cells were infected with the BCG MOHO. And what we see is a trend in the neonates, from cord blood cells from neonates, in IL-1 beta, would be the first idea that we had about this kind of cell death using detecting probability IL-1 beta, is that maybe what we have seen is not only necrosis, but could be also pyroptosis, but we haven't seen too much, we haven't advanced too much in that field to check what kind of cell death is happening in the neonates. So there are some features now, some characteristics of this study. One of them, and most important one, is that BCG is able to induce distinct cell death pattern during the maturation immune system. Remember that we use different cohorts to see that. And this pattern could set a stage for a subsequent antibacterial immune response, and this may have a profound effect during the vaccination. Having established that BCG MOHO is the strain that we have seen to induce such a different in terms of cell death, looking at different cohorts in adults and neonates, we are interested in see if other strains also can have this characteristic, this feature. So for that, we start using strains that belongs to the late strains, so Danish and Pasteur. And more recently, we start also studying Russia, our vitro. So we just repeat what we have done so far using the two cohorts, and the same way that we infect the cells with BCG MOHO. And what we have seen both in adults and neonates, that again, we corroborate the previous data showing that it was able to produce high levels of cell death, but we could not see the same rate. Actually, we couldn't see any kind of cell death induction when you used Pasteur or Danish strains. On the other hand, when you look at the cytokines, what we've seen, particularly the pro-inflammatory cytokines, is that the three strains that we have used so far, they were able to induce similar levels, let's say, of a pro-inflammatory cytokine, but once again, MOHO was the only that induced a lower level of TGF-beta, which is say, anti-inflammatory cytokine. So after establishing that is a different in terms of the strains, we also start using the Russia strain to see if there is any kind of difference in terms of cell death induction. Remember that the Russia belongs to the same group as MOHO. It's an early strain that belongs, it's a very similar to the original Pasteur strain was developed. But for our surprise, there is no induction of cell death when you look at the Russia strain in our model, now using the THP1 monocytes. But we again were able to see that monocytes, those lines were able to die easily by apoptosis. Then infecting the PBMCs from adults with those four strains, once again, we're able to see that once again was able to induce high levels of only pro-inflammatory, but also not only pro-inflammatory, but reduced level of TGF-beta, but more strikingly was the lower levels, particularly lower levels of IL-17 and interferon gamma that the BCG Russia was not able to use. And finally, we have, because of the cell-death pathway, we want to see if there is any kind of difference in terms of the candidates that would regulate the cell death. So we use a transcription model. You just check the levels of RNA and messenger RNA in those strains, in those individuals, adults that were infected with those two strains. And what we can see is that mostly BCG-MOHO was able to induce not only pro-apoptotic, but also anti-apoptotic candidates. But more important thing is that BCG Russia was not able to induce any kind of pro-apoptotic or anti-apoptotic gene expression. So there are some lessons, some issues that I want to enforce from the comparative study that we just made. So one of the things that I want to state that the BCG-MOHO, the strain is able to induce apoptosis in those already sensitized adult individuals. It seems to be more efficacious than Russia, Pasteur, and Danish strain. On the other hand, there was one study that was done using Tokyo strain. Tokyo also belongs to the group 1. It's an early shared strain. And Taniguchi and colleagues, what they saw is that Tokyo was able to induce prominent Th1 cytokine production in vivo in mice model, but we cannot share this data because we are not using Tokyo strain in our model. But one way or another, not all the early shared strain they are able to induce Th1 cytokine, since in vitro Russia was not able to do so. On the other hand, when you give BCG vaccine early in life, it seems to confer better protection against tuberculosis. It could be better evaluated in other trials. The MOHO strain induces different in vitro regulatory and pro-inflammatory cytokines when compared to Russia, Pasteur, and Danish. As I told you, we haven't done any study using the Tokyo to see if that could be also reproducible, as we haven't seen before. And as a whole, the results, our result reinforce the suggestion that MOHO is a more potent immune-stimulating agent than Russia, Pasteur, and BCG. And I put this sentence in a question mark because this could be attributed partially to methoxymicolate. However, Russia also belongs to the same group that produced methoxymicolate, and what we have seen is not a statement. So it should be careful, look at more carefully in terms of if really methoxymicolate is the one that gives this kind of different vaccine. So I'd like to give opportunity to my colleague Thales Campello, my Professor Campello, to talk a little bit about the prediction epitope study that he has done. And at the end of his talk, I will come back, we can wrap up and start the section. Thales.
Okay. Thanks, Dr. Paulo.
May I start?
May I start?
Yeah, you can go ahead.
Sim.
Oh, okay. My name is Thales Alves Campello, and I am PhD student at the Federal University of Ceará, located in the northeast state of Brazil. The first question about our presentation is, SARS-CoV-2 is still a problem? Let's examine what the epidemiological data tell us. Next slide. Slides. According to data from Ministry of Health Brazil, Brazil had 196,460 new cases of COVID during the period from January to February. So we question the reason behind this increase in disease case. Next slide. Studies indicate that the emergence of new variants leads to a decrease of vaccine efficacy. Next. please. So think about that. Our group has considered developing a recombinant BCG vaccine contains epitopes of SARS-CoV-2 and aimed to enhance protection against new variants of COVID-19. Oh,
20.
Next slide. So how does this process work? Firstly, we conduct the sequence of the protein sequence of the variants of interest. Followed by, we analyze bioinformatics of select peptides, and the finish we modeling molecular and recombinant BCG. Next, please. The first, find the complete sequence and SARS-CoV-2 in the website NCBI and UNPROT and PGB. The next step, we analyze the vaccine series about the antigenic peptides, and the followed by, we analyze immunogenicity, toxicity, and the finish, we analyze the interferon gamma prediction. Next, please. So in this slide, we observed an example about the sequence of the nucleocapsid region of SARS-CoV-2 variant. Next slide, please. In this slide, we analyze the vaccine and about the antigenicity. Next slide. And this slide, we analyze the check prediction of peptide binding with MHC class 1. Next. In this slide, we verify the antigenicity of the epitopes in question about the variants. Next. In this slide, we analyze the toxicity of the epitopes and the immunogenicity epitopes. Next. And the finish, we check the possibility of the interferon gamma prediction. And the final result about 2 peptides about this is analyze. Next step. So in this slide, we showed the roadmap about this is began with selection the peptide SARS-CoV-2. The next step, the peptide synthesis and the airspot assay. We are here. And the next step, extendation of gene recombinant assay BCG. For the next step, we initial of preclinical trials in animal models and finish initial of preclinical trials human models. The next. And thank you for your attention.
Yeah, thank you, Professor Campello, for your brief overview. I just want to make sure that what we have done so far, we have selected, I think, 5 peptides with using that approach that Thales just briefly gave you an overview. And we are now in the step of testing those 5 peptides using the cohort that I just showed you, cohort that adults that was recruited, enrolled before COVID, and also another cohort of adults that were recruited are still being recruited after the pandemic. So it's still a work in progress that since the beginning. So let me wrap up this talk with this. I'm very provocative in terms of using other strains of the BCG. One of the things that I want to call your attention is about, I just made a selection of papers, a screening of papers 10 years ago, more than 10 years ago, and I came up with the idea that there is only few papers that have been shown using different strains, particularly strains from the most immunogenic group. So one of the things that I came up with this review that I published is that there is a relationship between the recombinant BCG vaccine that are used in trials nowadays, and there is no study that I'm seeing so far that they have studied different parental strain, different construction to compare head and head to see even if you use different strain using that kind of recombinant that you induce in the BCG, you could have different levels of protection. In other words, what you can see is that only 2 of the 21 recombinant strains that I counted 10 years ago, they came from the most immunogenic group. They one study that revealed difference in terms of, one study that created one recombinant strain using the Moho strain, and the other one using the Tokyo strain. There are different constructs, but they use a different background. And it's still to come to see if that's construct, if you could be using in another strain, like a strain from the group 2 or group 3, the outcome would be the same. So I'm very provocative in the audience because I want to hear from them. If we using different constructs in different strains, what's going to be the outcome? I haven't seen any kind of paper comparing head and head. So to finish, what I want to focus on and to stress here is that what we want in the validate colleagues, is validate initiative is to build a group that can think about the future, particularly strategic, future and strategic partnership. And what we want with this collaboration is to bridge a gap between basic science and medical development. And I'm sure that the people will benefit from contributing impressive movement of science in Brazil using this kind of partnership. And I went to the Validate homepage and I took, on purpose, I took this sentence because I think that was interesting to remember that by working together, we advance research more quickly and effectively. That's what I expect. So what we want from this presentation, Professor Campillo and I, that we are deciding to launch a comprehensive partnership program with colleagues from Validate. This is to work hand in hand for environmental discovery. Our idea is to, together, we can advance our understanding in BCG vaccine using human model to uncover critical aspects of dysentery. So this work will not be possible, of course. There are a lot of people to acknowledge. I cannot put all of them here. So if I forgot one of them, please excuse me. All of them are important to make this work possible. 3 of them I just pointed out here is Carlos, Caio, and Andreon. They conduct mostly of the in vitro study in the lab. Professor Cartelo Branco is the one that is the BCG Moho provider from the FAP institution in Brazil that produced the vaccine. Cynthia Fernanda for the technical help since then, and also Paulo, Rafael, and Cristiano Frota from Ceará to their incredible collaboration that we have with them, and I'm very thankful. So also our support, the state agency and also national agencies give support to this study, and also the institution. I will leave here with you our email because I was talking to Blackley. Maybe some of you cannot stay in the Q&A question. We will be very, very happy to have a feedback from you, and very looking forward to hearing from you about this presentation. Thank you for your time. And Blackley, thank you so much.
Yes, thank you very much. Thank you, Paulo. Thank you, Thalas. And I'm just going to hand over now to the people in the Zoom. So, do we have any questions or just comments? Because it was quite. I think Paulo is interested in an exchange. So, do we have any comments from anybody in the Zoom? And again, you can chat. You can type in the chat as well. Okay, Isaac, please turn on either type or turn on your microphone and
All right. So, Professor, thank you for your presentation. It was very insightful. I'm talking from Ghana. I think from 2007 and 2008, the BCG vaccine strain that was used in Ghana was the Danish strain, and much recently they use a strain they call the BCG Japan. I don't know whether you have come across that strain. What can you say about the immunogenicity of the BCG Japan, if you are familiar with that particular strain? Well, so that's basically my question. Thank you.
Yeah, thank you for asking. Yeah, I would love to have that strain here to work with. It's one of the most immunogenic ones, not only because it belongs to the group 1, as I showed you before. Although belonging to group 1 doesn't mean too much because see, Russia is the same group. But what I've seen, most of the study that came from people using BCG Japan is that, yeah, there is a good immune response. However, good immune response doesn't mean that the vaccine is completely protected, or you can think about the side effects of the vaccine. What I can tell you, I'm not too much familiar with BCG at Tokyo. What I can tell you about that side effect when they use such a strain is that, particularly from the Moho, you have a lot of side effects, particularly in kids, when you give this kind of strain as a vaccine. So you have a good immune response, but on the other hand, you also have to deal with this kind of problem, let's say, that is the side effects that the vaccine has to give. But I don't think it's a very severe side effect. I don't think it's going to be something that is something to deal with in the clinical or maybe in the hospital. It's something that I'm talking is that there is, in terms of the scar, you can see a difference. It's bigger, much bigger, and also the way that the vaccinee, when we got this Tokyo vaccine, it's a little bit different from those that could receive a vaccine that's not as protective, let's say, like Danish or maybe the Pasteur. So what I can tell you about that is that it needs to be done. What needs to be done is that it should be done. Actually, comparing, as I told you, as we have done in Brazil, comparing the Danish that had been used in Uganda. Sorry, I think you are from Uganda, but what using in Uganda and now the Japanese strain, that should be compared not only using the model that I just showed in vitro model, but also clinical trial. That should be something that could be considered.
Thank you.
Okay.
Carry on. Sorry, I'm interrupting. Carry on.
All right. So as a follow-up, well, a correction, and I'm not in Uganda, I'm in Ghana. So I was referring to Ghana, not Uganda. But are you familiar with any adverse effects, severe adverse effects with the use of the BCG Japan, if you are familiar with any of them? And well, the reason why I'm asking this is because in my PhD work, we took evidence of scarring as an evidence of vaccination in some populations. And in Ghana, BCG vaccination is very extensive. We have about 98% coverage. But we realized from our study that a lot of people who claim to have been vaccinated did not have scar to show for it. Meanwhile, the data that we have from the ministry, we were expecting that most of the people would have had an exposure to BCG probably during their childhood vaccination. So I wanted to know if even with the BCG Japan, there are instances where you might not have scarring, as you showed for the BCG Danish and Russia, the BCG Russia. Are there any evidence like that you have come across?
Yeah, it's actually confusing. So Ghana, I was not aware that Ghana had changed the strain of the vaccine. I'd like to open your question to the other colleagues in the audience. I'm not MD, I'm not familiarized with the side effects. What I heard is directly from literature. I would like to hear if there's someone in the audience that can answer the question that you just made about the side effects that would be given by the BCG in Japan. I'm not aware of it. Sorry.
All right, thank you.
We have a hand raised from Dr. Thank God. Do you have a question?
Okay. Thank you, Paulo and Campello. A beautiful presentation. I must commend both of you. Sometimes I wonder, even to date, you see so many literatures. They tell you viruses are non-living things, while some tell you viruses are living things. So I've tried to explain to my students that viruses, I will not tell you because it's a question that I'm asking you, so I expect you to give me the answer before I tell you my own opinion. So I want to know, viruses, are they living things or they are not living things? That's one question. The second question is this: because of the nature of viruses, you mentioned COVID-19 against BCG. Is there not going to be antigenic drift and antigenic shift because of the nature of viruses?
The first question, I am biologist. What I could say to you is that they are not living things because they cannot reproduce by themselves. They need to enter in the whole cell, and to do so, they harm the cell, and then the way they can reproduce.
I disagree with you. I disagree with you that they, if you say they are not living things, what now gave them the life again when they now get into a living thing? When you say they are not living things, what now make them to live when they now enter a living thing?
I think, thank God, this might be a little bit outside of the scope of the presentation. So I think maybe move on to question two.
It's more philosophical than biological question, what I would say. So I'm sorry, I cannot answer that. I cannot answer the question.
Okay. So what about my second question?
So you ask about the BCG.
Yes.
Is there any
the
kind of
antigenic drift and antigenic shift?
It's a cross reactivity. What do you mean?
Yes.
So what? Yeah. So it's one of the things that I want to cover, but there is not too much time. One of the things that I told you guys when I presented the data about the agnostic effect of the BCG is that there are two main probabilities. One is the trained immunity, the other is cross reactivity. What you are talking now is cross reactivity. You know, I have one slide. Let me see if luckily allow me. I don't know how much time we have, but one of the slides that I want to show you is exactly what you asked. Here we go. So the study about using BCG to protect against virus, particularly is heterologous protection, came from this study that was done in 2021, where they saw is that BCG was able to induce at least 8 clones of T cells that could react against SARS-CoV-2.
Okay.
So 8 of the clones that when you vaccinate someone with BCG, 8 of the clones of the T cells, they cross-react with at least 8 antigens. What I could understand from the study, that are called peptide homologous. They came from SARS-CoV-2.
Okay.
So I don't know if that answers your question. So
yes.
This is one of the things that I want to show you, but
yes.
I'm not too much familiar with the study. So this is one of the study that gave us insight about why not using BCG against COVID. But as I showed you before, one study, a recent study, showed there is no effect using BCG against COVID-19.
Okay, thank you.
Thank you, thank God. Do we have any other questions? I think, Eamon, I think you turned your camera on. Did you want to ask a question? Or a comment? Because I think if nobody wants to speak up, then we've had some good questions there. nobody wants to speak up, I think that'd be quite a nice time. Oh,
Hello.
hi there. Oh, go ahead, Daniel. Go ahead, ask a
Okay.
question, comment.
Okay. Okay. Thank you, Black Ali, and thank you very much all the presenters for their wonderful presentation. This is really insightful. I have actually some questions or clarification. We know that TB is a complex disease, and the immunity is still not well understood in general. But when we study the immunity of a BCG vaccine or in general the TB immunology, strain variation in terms of inducing immunity in the host, there is a great variation. Strains are very variable, even in the BCG strains that are used for vaccine. From this study, we observe that the Murayo BCG induce higher apoptosis than the other. Is it due to, or have you considered the host pathogen co-evolution, that there are strains that have a sympatric host pathogen association and there are allopathic host pathogen association? Is there any difference in terms of inducing immunity based on their host pathogen association, and have you considered that? That is one question. The other is your strain, probably the Murao BCG strain, induced high apoptosis than the other. And what is the likely reason? Is it in terms of the strain preparation, or is it in terms of the host pathogen? I'm not clear why that difference was observed. Otherwise, it was insightful, and thank you.
Yeah, thank you for the question, Daniel. Let me start from the end. After 50 years of culturing BCG in different labs, what WHO did is that they are gonna make seed lots to avoid more differentiation in terms of genetic and biochemical. So after that, they create some guidelines to be followed. So I'm not completely aware about if those guidelines are the same nowadays for production of the vaccine all over the world. It doesn't matter if you use in different strains. I'm not aware about that. But what I can tell you from your question is that we are concerned about the method that we use to check for apoptosis. So what I can tell you is that we tested all the possibilities, including using the vehicle itself from different lots of the vaccines. If the vehicle could give a difference in terms of cell death, could be an explanation. No vehicle, no difference, so everything is going to be the same. For the 3 strain that we use, there is no difference in terms of the preparation as I'm aware about. I'm not sure about the process before that. So what we have seen is that one of the things that came to my mind when we start using BCG, live BCG, to infect the cells, that maybe the rate of cell death of bacteria, the died bacteria could be different among the lots. So we have seen in one of the papers, we show you that, I can give you the paper later, is that when we tested the bacteria itself, the vaccine itself, in terms of viability, they are the same. So the way that we reconstitute, the way that we infect, is what it was reproducible, it was the same, using that kind of method, the kind of staining that we did. So that rule out, in my mind, that rule out the possibility of different in terms of preparation of the strain after when the vial came to the lab. So I don't know before that. So what I'm trying to propose in this talk, as I think I want to make it clear, is that it should be tested more than what what we have done so far. So using the 4 strain that have been used so far is not enough to characterize that. So as I told you, I want to test other kind of strain, particularly from different groups in the genealogic tree. And also not only that strain that is the parental strain, but also their recombinant strain. Because as I told you, maybe when you make a different strain in terms of introducing or including another gene, you can also have a phenotypic difference in terms of the parental strain. So I'm not sure if that has been by 10 years ago when I start to make like a screen in the literature, it haven't been done so far. So I don't know if so I don't know if I asked your second question. So the first question that I don't remember exactly what you asked. Could you please repeat it?
Maybe the difference is this due to the host pathogen coevolution, that whether the strains are sympatric or allopathic association with the host.
Yeah, this one of the proposal that I'm currently doing with a colleague of mine. We are pretty much able to test not only several strains of the BCG, but also other species of mycobacteria, like using M. leprae, using Mycobacterium tuberculosis to see if when you go ahead in terms of virulence, you also have, I'm pretty sure that we're going to find difference in terms of not only biomarker, but also in terms of cell death. I would like to have a better tool to test that, Daniel. I would like to have a better tool to test not only the dichotomy between apoptosis and necrosis. No, I want to test better ways, the way the whole cell is dying. But so far, I don't have enough resources to do so. But one of the things that I want to, I told you that I'm very provocative. One of the things that I want to do in this talk is also to see if there is someone that is listening to me, if there is any kind of tool that I can use, we could use in collaboration to test this possibility, this hypothesis. So not only the one that you just told us about difference in terms of virulence of the bacteria, but other kind of question that is still popping up in my mind about difference in terms of the strain, but different in terms of the species, and so on. So and so if I completely answer your question, what I thought is that there is still difference in terms of virulence inside the BCG vaccine itself. Like I'm pretty sure that the same in terms of the pathogen itself. What we have seen, I think we follow in the literature, the Beijing strain of the MTB is much more virulent than other strains. So why that happened? I'm not following the literature in terms of comparison of the different grades in TB, but I'm pretty much sure that it happens also in any kind of organism.
Okay, thank you.
Yes, thank you. I give you a question, Daniel. Yeah, so I'm going to give people 10 seconds to quickly ask a question, but I think this would be a nice time to wrap up because the sun's going down here in the UK, so it'd be nice to finish up for the day. So quickly, any questions? Great. So I would just like to say thank you to Paolo and thank you to Thalas for a very, very interesting talk. And as you can see on the screen there, you can email either Paolo or Thalas if you want to continue this conversation. Yeah, do let us know. Validate can help with that. Maybe we could host a Zoom meeting or something like that. So do get in touch with them and me. Also, I popped into the chat a link to our joining page. So if you're not a member, there's a few non-members there. Do get in touch and go to that page and you can from there you can join Validate. It's free to join and there's many benefits which you can find out on our website. So thank you very much. And I'm going to stop the recording now. And yeah, I think we're gonna end it there. Thank you very much.